This is Kristen Haberther welcoming you to this Bite Size Bio webinar, which today is sponsored by Refine. Refine Limited spun out of Oxford University in 2018 with the goal to make the approach of weighing molecules with light available to a broader scientific community. Since then, they've gathered an enthusiastic team of people to develop, build, and support their mass photometry instruments, and they're closely working with their early adopters across the globe to further mature the technology. Today's presentation is titled How to Weigh Molecules with Light with Mass Photometry and is being presented by Professor Philipp Kukura from the University of Oxford. Dr. Kukura is the founder and director of Refine and full professor of chemistry at the University of Oxford. His research focuses on the development and application of novel optical methodologies aimed at revealing structure, energetics, and dynamics, and nanoscopic and molecular length scales. He has received support from the ERC in the form of starting and consolidator grants and has received awards from the Royal Society of Chemistry, the Harrison Meldola and Marlowe specifically, the European Biophysical Society and the Royal Society, as well as being awarded the Klug Wilhelmy Science Award and the UK Blavatnik Award. As always, we will have a question and answer session after the presentation. So please type in any questions that you have into the questions box, which will appear at the right-hand side of your screen. And I'll put them to Philip at the very end. So the recording of the webinar is going to be available at bitesizebio.com underneath our webinar pull-down menu, probably in about 24 hours. So now over to you, Philip, for the presentation.
Thank you very much for the introduction. I'm very excited to be here and to be able to tell you about the technology that we've developed in my group over the last decade or so. The concept behind this technology is really very simple, and it's illustrated by this rather trivial diagram that you see here on the front page. And the idea is the following. So suppose that had a single molecule in solution, and you would shine light on that molecule. And suppose that you were able to detect that shadow that comes off from that one molecule. Or alternatively speaking, you could detect the light that is scattered by that single molecule. Now, if you could do that, then you would probably agree with me that if you took two molecules, that shadow would be twice as strong. And if you took 6 molecules, that shadow would be 6 times as strong. And if you use every time the same molecule, then you might come to the conclusion that there is quite a good relationship between the strength of that shadow, or the strength of, or the amount of scattered light, and the mass of the species that you're looking at. And that's really the basic concept behind what we do and the technology that we've developed. We look at molecules, we shine light at them, we look at the light that comes back, and by quantifying the light that comes back, we can actually determine their mass. And this is where the concept of weighing molecules with light comes from. So the reason why such a technology might be a good idea is really quite simple, and comes down to the fact that if you take pretty much any process in nature, you can break it down to molecules coming together and falling apart in space and time. The example that I've chosen here is tubulin, which goes from being dissolved in solution to forming microtubules and back. And this process is fundamental to things such as cell division, as shown in the image on top. What the cell does is it builds up this framework of microtubules, and this framework helps distribute the genetic code between daughter cells precisely. So every time this happens, you go from solubilized tubulin to microtubules, build up the network, and then the whole thing falls apart again, and you go back and forth. Now, what we've become quite good at is getting atomic resolution pictures of the proteins themselves. But where life gets quite tricky is being able to really find out more about the dynamics as to how you go from, let's say in this case, the solubilized version to the tubules and back. Now, what I've shown here is how we tend to think about these things. So very often we will draw blobs and blobs and arrows and things going back and forth. So this is how we conceptualize, how we understand the underlying mechanisms. But measuring these things and quantifying them is really quite a challenge. And the underlying problem here is that ultimately, what you would really like to do is you'd like to be able to actually see this process happening in real time, follow individual molecules as they go about their job, in this particular case, from tubular dimers to microtubules and back. Now, you may say that there is not, what's the big problem here? Because we are able to see single molecules and have been able to see single molecules for a long time, since the late 80s. And indeed, single molecule optics and our ability to apply it to the life sciences has had a really dramatic impact on the kind of experiments and studies that we can do. Now, the huge advantages that you have there are that by using fluorescence, so using the fact that you shine light at one color on your sample and detecting light at a different color, is that you can visualize specifically the molecules that you're interested in, either because they are the only ones that are fluorescent or because they are the only ones that you've actually made fluorescent. And you can really generate very beautiful high contrast images of single molecules, such as the one that I've shown you above. So the big advantage here is you are single molecule sensitive and you are specific. If you wanted to find the downside of this approach, it would be that it's not universal. So it will not work with all molecules. It will not work with all proteins. In fact, it won't work with most of them. It will only work with the ones that are fluorescent or the ones that you make fluorescent. So this approach is not going to be universal. If you are after something that is universal, you have two main options. One is to use light scattering. This is what you use when you look at X-ray crystallography or electron microscopy. You shine electromagnetic radiations or electrons on your sample and you look at the scattered light that comes off. And matter interacts with light, so it doesn't really matter which protein you choose or biomolecule you choose. Assuming everything else behaves nicely, you will get something like a nice structure shown on the bottom right. The other option is to use mass. And of course, the big, huge advantage of using mass is that it is specific. One biomolecule will have a slightly different mass than a second biomolecule, and therefore you can find out about interactions, you can find out about structure, and you can find out about dynamics. Now there, you have that advantage that both of these approaches are universal and they are specific, but you don't have that single molecule aspect. You don't have that ability to look at what happens with individual molecules and what happens as a function of time. So our challenge really has been to try and square this circle, or more of a triangle, and see whether we can take advantage of the unique capabilities of single molecule techniques in a way that is universally applicable. So the big question then becomes, how might you take advantage of the single molecule capabilities, but still do it in a universal and specific way as enabled by measuring mass or light scattering? And if you look at this slide, there is a clue to how this might work, because one of the most popular approaches to visualizing and studying single molecules is to use light. In this particular case, to use fluorescence. And a universal way of looking at molecules is to take advantage of light scattering. So the most obvious way might be to say, well, one uses light, the other one uses light. Why not combine the two? So why not use light scattering? Well, what's going to matter here is how strongly or how weakly your light is going to interact with the object of interest. So that may be your individual protein in this particular case. Because, of course, it's one thing to think about how this might work, and it's another thing to actually be able to do it. Now, the interaction strength is governed by the cross-section, which is essentially the effective size of your object or of your biomolecule when it comes to the incident light. And that is roughly proportional, as shown in the equation above, to the square of the volume of the object and the difference in refractive index between the object and the medium. Now, the first term makes sense. The bigger, the more signal you get, the smaller, the less signal you get. And the second term also makes sense. You'd expect a piece of metal to scatter a lot of light in water, but you wouldn't expect a drop of water to scatter a lot of light when it's immersed in water. Now, then the question becomes, is this actually experimentally feasible? And when we started this, you could look into the literature and you could go and search and find the most sensitive microscope when it comes to detecting scattered light. And what you would find is that the detection limit, the smallest thing that you could see in such a microscope, was something on the order of a 20 nanometer gold particle. Why gold? Well, because gold is really good at scattering light. So you can quote a really small number when it comes to the size of the object. But then you could do another calculation and say, okay, if that's the smallest thing I can see, what about a single protein? And you would very quickly come to the conclusion that a single 20 nanometer gold particle scatters about 1 million times more photons than the single protein. But now you have a big problem because, of course, what you find is that the best microscope you can buy lacks sensitivity by a factor of 1 million just to actually see the protein. And that's another challenge then to be able to actually quantify the amount of light that comes from it. So at this point, the only smart thing to do would be to just give up, because maybe 1 or 2 orders of magnitude might be something that you could envision working against, but 6 or 7 or 8 orders of magnitude are really going to be quite a big problem. Now, what we've done is to some degree just ignore this problem and look at it from a slightly different perspective. So here's the idea. So let's say you just asked a very simple question. I shine light on a sample. I look at the light that comes back. What's the smallest thing I can see? So I build a microscope that looks a little bit like the one that you see on the top. You have a laser light source. You point it towards the microscope objective, goes towards the sample. You look at the light that comes back and you put it on a camera. And if you build a microscope like that, what you will see on your camera is shown on the bottom left, which might be the explanation why nobody's really pursued this particular approach. Because what you find after you spend 20,000 pounds and several months of work, or maybe several weeks of work, is that all you've managed to do is take an image of your laser beam profile. So that's not particularly exciting. And you will also find that the intensity varies by about 90% across that image. But what you're trying to see is a tiny, tiny, tiny signal that you might get from a single molecule. Now, one of the things that you may realize at this point is, okay, so it's not great that I only get an image of my laser, but that's not really what I'm after. What I'm after is seeing a difference. So I can measure that laser beam very, very accurately, and I can just subtract it from any other image I take from my sample. And that's what we did in the middle. So if you just take a cover slip, immersed in water, you put it on top of your microscope, put it in focus, and you subtract the background that you get from your laser, you get what you see in the middle. Now, if you look at the scale on top, it says reflected intensity. So what you can see is that you get something that looks like a speckle pattern, kind of undulations in reflectivity. And those reflectivity differences are on the order of a few percent. All right, so that's not particularly exciting. You've taken an image of the reflectivity of your cover slip. But then the next question is, how big or small is that signal? What does the signal actually correspond to? And the answer is on the bottom right. Basically, it's actin. So if you take actin, which is another cytoskeletal protein, a filament, and you just put it down on a cover slip like this, you see the image that you see on the bottom right. And you can just about see thin, faint lines, dark lines that kind of crisscross across the image, maybe with a signal to noise ratio or a visibility of 2 or 3. Now, what does that mean? Well, we know the structure of actin really quite well. We know what the diffraction limit is of our microscope, so what the tightest focus is that we can achieve with visible radiation. And so we can calculate how many molecules actually contribute to that signal that makes it a very faint line in an area of, say, 200 nanometers. And when you do all of these calculations, you find out that this very, very simple microscope has a visibility limit of about 70 proteins. So the line that I see corresponds to the signal that I get from 70 proteins that are attached to the surface at that point. So there are two reactions to this result. The first one is, well, that's amazing. I thought I was a factor of 1,000,000 off, and now you're just telling me you're a factor of 70 off, so that's a good result. The second reaction is, well, congratulations, you can see 70 proteins, but you can't see one, because of course your background is way too big. Now, this is where my daughter comes into play in the sense of these books here. So I'm sure many of you will have witnessed these wonderful things at some point, usually late in the evening when everyone's exhausted. So this is Where's Wally or Where's Waldo, depending on what country you're in. And the concept of these books is very simple. So you see Wally on the postcard there on the top, and Wally has friends, Odlo and the Wizard and all kinds of other friends. And there's also wonderful objects such as keys and bones, all hidden throughout these kind of images that you see here. Now, if you think about it, who we're looking for here is Odlo. So you see Odlo at the top. Now, finding Odlo in this image isn't actually that different from trying to find a single protein in this image here on the bottom right. It's essentially the same challenge, right? There's lots of stuff going on, and you're trying to find a tiny signal. So I could now ask you to try and find Odlo, and it would take you a while. Might take you forever, depending on how good you are at this, but it certainly will take you a while, and the reason is because you have a lot of background. But there is a trick here that you could use that would make it very, very easy to find Odlo, and the trick is you would take one image with Odlo and one image without Odlo. So this is with Odlo, and this is without Odlo. With Odlo, without Odlo, with Odlo, without Odlo. And you can immediately see where Odlo is. And in fact, what you now can do is you could subtract an image with Odlo from the image without Odlo, and all you end up with is Odlo. And that's essentially exactly what we've done. So on the bottom left, you see again these faint lines due to actin being stuck to glass. And now what we've done is we've added a molecular motor called myosin 5, which binds to actin. And if you add ATP, it will bind and walk for a little bit and then unbind again. And then another one will come from solution and bind, walk a little bit, and unbind again. So what we did is we took this image, then we took a video, added the motor and ATP, and just subtracted the first image from the ones that came afterwards. And the movie that we got is the one that you see in the middle. So wherever you see those faint lines, you see little black dots that arrive, and then they move a little bit, and arrive and move a little bit, and some of them leave. And when you get really lucky, you get a very high signal to noise ratio observation, and you see something like what you see on the right. You see this black dot, and you look very closely, it actually takes what look like digital steps. And those digital steps are about 37 nanometers, and that is the step size of this motor. So we had pretty good evidence that indeed what we're seeing here, in fact, are single unlabeled biomolecules, in this particular case, molecular motors. So there are no labels, there's no dyes, there's no gold particles, there's no enhancement, there's no nothing. It's just shining light on the sample and looking at what comes back, subtracting the background, and indeed we can now detect, we can image, and we can track individual proteins in solution. Now you will look at this, and maybe you'll think back to the slide that I showed you with single molecule fluorescence, and you'll say, okay, that's great, but actually those fluorescence images, they looked a lot better. Why are you doing this? You know, what's the point? And this is where we return to the original challenge, right? You're absolutely right. The fluorescence images look sharper, look prettier, but you're lacking that universality. Now, if you look at the way that we detect our signal, which in this particular case is a combination of light that is scattered by the molecules and the light that is reflected at the interface, in this particular case, glass and water, that signal is proportional to the polarizability of the object. And as shown in the equation on the right, that polarizability is proportional to the volume, and again, the difference in the optical properties between your object and your medium. So you have an advantage here in that your signal is no longer so strongly dependent on volume, so it doesn't drop as the volume squared, but linearly, so you have a slightly weaker drop-off in signal. But what you find if you look at we did this here for 170,000 proteins from different organisms, is that actually the optical properties of proteins are all pretty similar. And that's not that surprising because they're all made of the same amino acids. It's not like you have some proteins that are made of metal and others that are made of amino acids. They're all made out of amino acids. And the other thing that you realize is that actually the specific volume is pretty much the same. So in other words, the density of proteins is pretty similar. And you know this because there aren't that many 50 kilodalton proteins that are 2 nanometers across and some others that are 20 nanometers across. They're all pretty much the same size. Or saying that better, they all pretty much have the same density. So what that tells you is the second term in that equation is pretty much constant, and the first term of in that equation is proportional to mass. And now everything comes together, right? Because we can see single molecules. We can do it in a universal fashion because we're detecting scattering, not fluorescence. And that scattering signal that we detect is directly proportional to the mass. So there is one issue, however. It's nice and well that in principle this works, but if you looked at that movie that I showed you before, by my own admission, as much as I like that data, you have to be a little bit squinty-eyed to see that protein. And in that particular case, myosin 5 is nice because it's 500 kilodalton, it's relatively large, so it actually produces a big signal. So that data was taken now 5 years ago, so we spent a lot of time really optimizing the experiment itself to try and get out as much information as we can in terms of the scattered light. Now, I won't bore you with the technical details, but I'll just show you the outcome of this. So here's a movie of HSP 16.5. It's a heat shock protein, 400 kilodalton in mass. And essentially the same experiment as before, except that now we're binding molecular motors to actin, but we're just doing non-specific binding of this heat shock protein to just a bare glass surface. Every time you see a black dot, that's a protein binding, and every time you see a white dot, that's a protein unbinding. Now, the fact that the black dot disappears is not because it unbinds, it's because of odd law. Remember, if you subtract an image without odd law from an image with odd law, you get odd law. But if you subtract 2 images with odd law from each other, you see nothing. And so when molecules get immobilized and you subtract them from each other, eventually you don't see anything. Now, I'm going to play this movie one more time. If you look at this long enough, you realize that curiously, pretty much every single one of those dots has exactly the same intensity, right? Now, if we take a different heat shock protein, one that is polydisperse, so HSP 27, now you don't have that luxury where every single object in your solution has exactly the same mass. And immediately you can see some of those dots are really, really faint. You can barely see them. And some of them are really, really strong. So that gives you an indication, or gave us an indication, that indeed this idea that our scattered signal is proportional to the mass, or in this particular case, how many copies there are for each of those individual binding events, holds true. Now, we took this idea and really pushed it to the limit, and the outcome of this is what we call mass photometry, or weighing molecules with light. So we take these movies, we record the signal. If you take something like BSA at a particular concentration in solution, you get these little dots that you see on the bottom left, the scatter plot. So depending on where the dot is on the right is proportional to how strong the signal is, and where it is on the left is how much time has passed. And you can immediately see that there are 4 regions. There is one around a contrast of 0.4, one around 0.8, 1.2, and so on. And if you integrate all of those up, you can see a large peak, a smaller one, an even smaller one, and a very small one. And they're all equally spaced. And the immediate conclusion from this is, well, the big one is a monomer, and the smaller one is a dimer, and the even smaller one is a trimer, and the one that you can barely see is a tetramer. Now, we've tested this then more specifically by looking at a whole bunch of different proteins and plotting the sequence mass versus the size of the signal that we actually measure in our instrument, which is shown in the middle. And you can see that, as expected, that relationship holds true. They are proportional to each other. And in fact, the relationship is very good. So when we take the difference between a fit to those points and what we would expect, the mass error that we get over a broad range from about 40 kilodalton to a megadalton is on the order of 2%. And that's actually quite good. If you look at native mass spec, that accuracy may be on the order of 0.5%. And if you look very closely at this graph, actually above 150 kilodalton, our accuracy is comparable to that native mass spec number. Now, we've done a few other checks. In yellow, you see we've tried to change the shape of the protein to see if that has a significant effect on the signal that we see. It doesn't appear to be the case, at least not that we can measure it. It turns out that if you take systems that are not just made of amino acids, and of which there are plenty that people are interested in, this approach also seems to work. So on the top right, you see some work we've done on lipid nanodiscs. So this is about 50-50 mass between amino acids and lipids. And not only do we measure a mass that is roughly along the expectations you would get from techniques like DLS, NMR, and mass spec, but more importantly, the changes that we see when the only thing we do is change the amount of lipids or the nature of the lipids is in line with the expected contrast changes. So it also seems to work for lipids. On the bottom in blue, you see a test for what happens when you have a protein that consists to about 30% in mass from sugars. And we changed the nature of the sugars. And again, the change in mass that we see corresponds to the expected occupancy of the sugars on the protein. And then finally, as a kind of an extreme test, we looked what's the smallest change we can see, what's the smallest difference in mass between two different samples. So we titrated biotin to streptavidin, and the smallest difference that we could see at the time is on the order of about 1 kilodalton. So approaching an ability to detect binding of small molecules. Now you might ask the question, didn't you just tell me the whole time that the only reason why this works is because amino acids are the same? And you're right, it is to some degree surprising that it also works, seems to work with lipids and sugars and small molecules. Now, my explanation is, and admittedly this is not perfect at this stage, but if you just look at a cell in a transmission light microscope, everything just looks like a blob, even though it has lots of different biomolecules in it. And the reason why it does is because, well, most of these biomolecules have the same optical properties. Again, they're just made out of the same organic material. They're not made out of metal. And therefore, this approximate relationship between the scattering and the mass that we have not only seems to hold very well for amino acids, but also holds very well for lipids and sugars. So the key question is, what can you do with this? And I just want to show you a few examples. And the reality of it is we're pretty much every day finding new applications for this approach. But I just want to show you a few examples, which I think kind of cover quite nicely the potential applications of this technology. So here's a very simple one. Is my protein pure or not? Right? So if you take something like HSP 16.5, I said it's a 400 kilodalton protein. It's not a single covalent chain of amino acids as 400 kilodalton. In fact, it's a 12-mer of dimers. So if you just purify that protein and you run a gel, you'll see, okay, here's a band at 16.5 kilodalton. But when you have it in solution, is everything really assembled as this one particular cage at 400 kilodalton, or do I have a range of different assembly intermediates? Which is kind of a similar question to how does something like this actually build up? How does it assemble, right? Is it just a pearl on a string where every association constant is the same, except for the very last one? Or do I make tetramers and the tetramers assemble into a 24-mer or something else? Now, because we're measuring the abundance of species as a function of mass, we can exactly start to answer these types of questions. So if you take something like HSP 16.5 at a relatively high concentration, well, depending on what you consider as high or low, but in the comparison here, 400 nanomolar, you see you get a wonderful, nice big peak around 400 kilodalton or a 24-mer. Now, when you start and dilute this down, you can see that this 24-mer peak drops. But not only that, at the same time, low molecular mass or low oligomeric species begin to grow in abundance. And the dashed line is a fit to a model that assumes that indeed every equilibrium constant is the same except for the very last one that locks the structure in place. So you can look at this in two ways. One is by measuring these distributions as a function of concentration, I can figure out what the KD is of my oligomeric species, and I can find out roughly as to what the likely assembly mechanism is, because if there were any intermediates, I would see them as a high abundant species in the spectra. The other way to look at it is if you want to study HSP 16.5 as it exists as a 24-mer, don't do it at 50 nanomolar, because at 50 nanomolar, only a fraction of your species is in that assembled form. Now, this is a relatively simple example in the sense that what we're really dealing with are interactions of molecules with themselves. Now, the next level of complexity would be to look at interactions of molecules between molecules, right? So here's a very simple situation where let's say you have an antibody and you have a protein that binds to the antibody. So you want to know, do these two molecules interact, yes or no? In the top left, you have a scenario where they do not interact. So if you were to run a mass photometry experiment, you would get two peaks. You would get a peak at the antibody mass, and you would get a peak at the protein mass. Now, if they were to interact, like shown on the top right, this situation would change. You would get less free protein and less free antibody, and instead, you would suddenly see a peak at the mass of the sum of the masses of the protein and the antibody. So here's a very simple example of actual experimental data that does exactly this. So in the absence of your protein that's supposed to bind to your antibody, which is the light blue line, you just get a peak at around 150 kilodalton, as you would expect for free antibody. As you titrate in your ligand, you see that the intensity of that peak drops. And if you put in lots of your ligand, then eventually you only get a peak at 200 kilodalton, which in this particular case is the sum of the mass of the antibody and its ligand. You can repeat that for different concentrations, and you can fit that to a curve in a traditional way, and what you get out is the specific binding constant for this particular interaction. Now, you can probably also guess that just by looking at these distributions, you can get a pretty good idea of what this equilibrium constant is just by looking at the distributions themselves. So let's look at a slightly more complex example, which would be, rather than a one-to-one interaction, something where there might be something a little bit more complicated going on. So the example that we looked at here is the interaction between HIV envelope protein and an inhibitor called BANLEC. It's a lectin. The means of action is that the lectin binds to the sugars which are on the envelope protein. And one of the questions is, okay, how does this actually work? How does this inhibition work? There are two possible ways that this could happen. They're shown on the bottom. One is the envelope protein is coated in sugars, the lectin coats the envelope protein and thereby inhibits the action of HIV. The other possibility is it cross-links and thereby aggregates all the envelope proteins on the surface of the virus and thereby inhibits. Now, if you're able to count the number of species and determine their mass in solution, then you can exactly start to address these types of questions. So if they don't interact, as you now know, you get two peaks, one at the mass of the envelope protein and one at the mass of the lectin. If the lectin were to coat the envelope protein, all you would get is a shift in mass. So because it just becomes heavier. Whereas if you were to cross-link, what would happen is you would lose monomeric envelope protein and you would get dimeric and trimeric and tetrameric and so on envelope protein, in addition to having a shift in the mass because you've added the mass of the lectin. And that is essentially exactly what we see. So what you see on the left are mass distributions measured at different concentration of the lectin. You titrate in the lectin, your monomeric envelope protein around 300 kilodalton drops, and your dimer around 600 increases, and your trimer increases, and your tetramer increases. You can convert these mass distributions into mole fractions. You can fit those mole fractions to a relatively simple model, and from that relatively simple model, you get the different binding constants. And from the different binding constants, you can determine the mechanism, which tells you indeed what's going on. There is some cross-linking going on. And one of the reasons why this works so well is because of cooperativity. In other words, a lectin bound to envelope protein is much more sticky than a lectin that's just in solution. So everything that we've talked about so far is just looking at distributions. one thing that would be nice to have a look at is to see how these distributions evolve as a function of time. So it's one thing to get a structure and something static. It's another thing to see how that changes. So what I'm showing you here is from a very recent paper just a few weeks ago that appeared in Nature, collaboration with Jonathan Heddle, and they are interested in making protein cages, and they're very good at determining the structure using cryo-EM, and you can see that structure in this particular image. But one of the questions that they had was, how do these structures actually form, and what are the kinetics of the formation? Now, this particular structure here would have a mass of 2.1 megadalton, whereas the individual subunits are 90 kilodalton. So what you would say is, well, why don't I just measure mass distribution as a function of time after initiating assembly? And that is exactly what we did. So on the bottom left, you can see the mass distributions as a function of time. You see after 30 seconds, already you have something that's at about half the mass, and then the mass shifts to about 2.16 megadalton, which is exactly what you would expect for this assembled protein cage. And because we can quantify the fraction of species at 2.1 megadalton, we can determine the kinetics of this process on the right. Now, in addition to that, what you can do is you can take the same solution and look at it in the absence of gold, which in this case induces assembly. And that is what is shown in the inset on the bottom right. And you see there's a peak at 90 kilodalton and 180 and 270 and maybe 360. And you will now probably guess that the subunit that assembles this cage is a 90 kilodalton ring, which eventually forms this large structure. So you can characterize where you're coming from, you can characterize where you end up, and you can do it as a function of time in a dynamic fashion. Now, everything I've been telling you about so far is about abundances, right? So how many species do I have at what mass, and how does that change maybe as a function of time or conditions? But the way we measure it is by taking actually images, such as the ones that you see here that make up this movie. Now, these kind of blinky images you probably have seen in the past 10 years or so, because they are quite reminiscent of what you would see if you're doing stochastic super resolution fluorescence microscopy. And one of the things you do there is you identify every single one of these molecules and you fit it and you find the center of mass. And from that, you can build up a super resolved picture. And your ability to localize these species really depends on the quality of the images that you get. Now, one thing that's quite remarkable here and worth mentioning is that you can see that the signal to noise ratio is very high. And in fact, it is so high that at about 10 hertz, our localization precision for this particular protein, 400 kilodalton protein, is sub-nanometer, which is something that's actually really quite difficult to achieve with fluorescence alone. Now, what that tells you is that not only can you measure distributions, but you can also do it in a spatially resolved manner. And I want to show you just one example of where that might be useful. So we looked at aggregation, right? So the idea is very simple. What it comes down to is, let's say you go from a situation where you have a homogeneously distributed sample to a situation where that sample is aggregated, or vice versa. And what you will agree with me is that locally, your refractive index will be different in the two scenarios. So what we've done here is we've looked at synuclein, we've added synuclein to a membrane, and one of the things that people expect to happen is that the synuclein will aggregate. But how do you actually monitor that process? Well, when we did that in our microscopes, what we saw is what you see in the movie on the bottom right. So we add the synuclein to an appropriately charged bilayer, and then within seconds, we see these little black dots appear. Right? And these little black dots are a consequence of the synuclein initially being homogeneously distributed and aggregating. And we can look at every individual one of these dots. We can measure how the contrast and or therefore the mass changes as a function of time, and we can do that as a function of the concentration of synuclein that we add to the bilayer. So from that, we can really begin to characterize aggregation processes, or vice versa. If you wanted to look at stuff falling apart, you would see exactly the opposite, only in terms of contrast. Now, if you take all of this together, I think there is a good case to look at this and accept the fact that in a way this is a new way of measuring mass, or maybe in this particular case it might be more appropriate to say to see mass, right? Because the way we've been doing it for thousands of years is in a mechanical fashion, using gravity as a foundation of today's society to some degree through trade. 100 years ago, the mass spectrometer was developed, where you take molecules and you put them in a vacuum, you charge them, you send them around the racetrack, and that's how you measure the mass. And now we have what we call mass photometry, where what you do is you just look at the molecules. You shine light on them, and from the light that comes back, you can determine their mass very accurately. One aspect of this that I think is always very important, in particular in the context of the types of applications that I mentioned, is how easy or difficult it is to do this experiment. Because it's one thing to show you, nice data and say, oh, we can characterize this and we can characterize that. But what you don't really know is, okay, can I just actually go and do this? Or do I need to be 4 floors on the ground on a 50-ton slab of concrete in a helium-filled room, and 1 out of 1,000 experiments works? So what I'd like to show you as a movie is how the experiment actually works. And so what you see here is essentially the procedure, and it's really quite simple. You take your material that you're interested in, you take your pipette, you take a few microliters of your solution. This is the microscope, a commercial version of it. You put a little droplet onto that microscope, you close your lid, you look at your screen, and there are your molecules. And these molecules in this particular case are about antibody sized. And what this movie shows you is 2 things that are important. One is how quickly you actually get visual feedback, direct feedback on what your sample looks like. Because if you remember my heat shock protein, you can see pretty quickly whether your sample is monodispersed or polydispersed without having to do any further analysis. Just your ability to look at these movies will tell you. And the second thing is, so this is, I think, on a beer bench in a convention hall. So there really isn't a lot of temperature control or vibration control or humidity control or any other control. It's just sitting there, and you can still do these very, very sensitive experiments. So to conclude, some of the kind of key applications that we're excited about and that we really think where this technology will be able to contribute quite a bit in the future. On the one hand, the obvious one is to any types of biomolecular interactions. It could be protein-protein, you could be interested in antibodies, you could be interested in sugars or nucleotides or lipids. Whenever you have a situation where you have 2 species with different mass, and when they combine, there's a new mass, in principle and in practice, as I've shown you, you can start to characterize exactly these kind of interactions. A simpler version of that is to just look at sample purity, concentration, and structure. Right. So if you take something like GrowEL, which is a 14-meric protein, 800 kilodalton, and you measure that at a low concentration where you expect it to fall apart a little bit, sure enough, you see a bunch of peaks. Around 800 kilodalton, which is the assembled species, 400 kilodalton, and then individual peaks around the monomer, dimer, trimer, and so on. So in this very, very simple mass distribution that you see here, you actually get a huge amount of information. It tells you whether your protein is actually assembled or not. It tells you what fraction is assembled. It tells you, to some degree, what the structure or the likely structure of your protein may be, just by being able to see what distributions you have. And then finally, as I've shown you with the example of the protein cages, by measuring these distributions as a function of time or as a function of conditions or temperature, you can start to measure kinetics and find out how things assemble or disassemble as a function of time. Which leaves me to say thank you to the people that actually did the work, because really all I did was to be annoying and tell them to do the experiments better. So this is a picture of my group and a few collaborators who are really the people who do all of this work. And I'd like to also highlight Justin Benesh, my collaborator, who's a native mass spectrometrist, who's really been instrumental at every level of making this technique what it is today and enabling the capabilities that we have today. My collaborators, the people who actually make this possible in the spirit of providing the funding that allows us to develop the technology. And as a final bit, just to disclose that I'm a founder and director of Refine Limited.
So Philip, there we have a couple of questions from the audience. There's 2 of them here from Heidi. Her first question is, what is the lower molecular weight cutoff for the technology to work?
Well, I think, I mean, there's 2 ways to answering this question, because in principle, it ultimately comes down to what signal to noise ratio you're comfortable with. In general, we don't tend to go below 40 kilodalton. So those are the smallest things that we can see. Now, have we managed to see signatures of smaller species in the lab? Yes. But I think a downside is if you try and go smaller at the moment, at least, is that you run the risk of not being so quantitative anymore.
Okay.
So the general answer is 40, but if your heart depends on going lower, it is possible, and we've done it.
All right. Fair enough. She also has a question. Does it only work for soluble proteins, or does it also work for membrane lipid associated proteins?
It's a very good question. in some ways, you kind of had the answer in that talk there. When you go all the way back to that mass photometry, you saw that we can look at lipid nanodiscs. And so the answer is really yes, it works. It works in exactly the same way, assuming, of course, that you've solubilized your protein. And then the question is, what do you solubilize it in? The kicker here is, of course, we see everything, right? So we see the protein and we see the lipids. And if you have something like a nanodisc, then you get very homogeneous. You tend to get very homogeneous samples because your nanodisc samples tend to be very homogeneous. But if you have a detergent, and if your detergent is very heterogeneous, then of course that's going to broaden these distributions. So we've done experiments with a whole breadth of membrane proteins, and it works. But the kind of information that you get is information about the protein itself and about the carrier, which you may either want or you may not want, depends on exactly what you're after.
Fair enough. There's another question. Have you tried this technology in live cell samples? Because it's two-part. So it's, have you tried this technology in live cell samples, and how would you differentiate different proteins or biomolecules in their assembly?
Okay, so live cells are probably not our forte. This is where fluorescence-based techniques really, come to the fore, and the reason is, with fluorescence, you have the advantage that you can tag one thing, and even though you have lots of cellular material and membranes and all kinds of stuff floating around, what the fluorescence will do for you is make sure that you only see the thing that you want to see. Since we are label-free, everything produces signal, and so when we look at cells, what we get is a very large signal from the cell and any fluctuations that may be going on. So we haven't really applied this to cells. Am I going to say that, it's completely out of the question, impossible? No, that probably would be unwise. But currently, the real strength of the technologies is when you're working in vitro.
Got it.
And the second bit was
how would you
differentiating different proteins.
Yeah.
So now, is that in a cellular environment or in general?
Let's. It doesn't
I think
specify.
in a cellular environment,
Let's just
I guess
cellular.
we covered that. That's not going to work.
Yeah.
In vitro, well, it all depends on the mass difference. So if you think back to the mass distribution that I showed you for GrowEL, that's kind of what you do, right? You can see there's a monomer and a dimer and a trimer and a 7-mer and a 14-mer. So you can differentiate species as a consequence of them having different mass. Now that, of course, will become increasingly more difficult if the species that you're trying to differentiate, if their mass becomes closer and closer and closer and closer. But so telling the difference between 50 and 50, differentiating a 50 and a 55 kilodalton protein in a mixture is tough.
Yeah.
But if you want to see a free antibody and an antibody bound to its antigen, and the difference is 40 kilodalton, then that's certainly something that we can do. So it really depends on the mass difference, to which degree you can distinguish them.
Got it.
In one go.
Yeah. Okay. Can you measure binding and unbinding kinetics in the millisecond time regime?
Ooh. Milliseconds. So then now, of course, the key question is: do you mean 1 millisecond or do you mean 100 milliseconds, right? So let me answer it this way: it's a little bit of a function of the mass of what you're trying to see. So let me give you an example that I think would be realistic. if you say you had a scenario where you have an antigen on the surface and you're interested in measuring and quantifying the binding and unbinding of antibodies to those surface immobilized antigens. Given the mass of the antigen, if all you want to know is binding and unbinding, certainly I would feel comfortable at the 10 millisecond time scale and potentially a little bit faster than that. So when your protein becomes smaller, then you become a little bit slower because you have to average a little bit more. But it gives you a rough idea of where we would expect to be in terms of binding kinetics.
Sure. All right. Excuse me. So this Scott says, your noise in your difference profiles looked like there was an evidence of trajectories. Can you sample faster than 10 hertz?
Could you, somehow you cut out halfway.
Oh, sorry. My apologies.
Just start this
Yeah.
one more time.
Your noise in your difference profiles looked like there was evidence of trajectories. Can you sample faster than 10 hertz?
Yes, I mean you can. The sampling rate really comes down to how much you want to suppress the noise. So for example, the experiments on the assembly of the protein cage, they were run, I believe, at 500 hertz or 1 kilohertz. And ultimately, your limitation is how fast will your camera go and how small is the object that you're trying to see. So you can go faster, and you can, in principle, certainly, right? I mean, the myosin movies were an extreme example of this. You can use it also to track objects move around. But there's always a trade-off, right? The smaller the object gets, the slower your time resolution. The bigger the object gets, the faster you can go.
Right. Okay. And what is the optimal protein amount and concentration to perform measurements?
So the happiest, there's a range, but if you wanted to make the completely straightforward experiment, I'd say 50 nanomolar. And when I say 50 nanomolar, I mean 50 nanomolar of object concentration. So this is where we think about concentration a little bit differently than you might traditionally. So if you have a 20 former, then 50 nanomolar would correspond to what did I do? 1.2 micromolar or something like that of the monomer, right? But it's what is the concentration of the object? And 50 nanomolar is about right. How much? That kind of depends on your skill. So the way we do it is usually several microliters, because that's just what you can pipette easily, and that's the kind of sample holds, gaskets or flow chambers that we use, 5 to 10 microliters. So really very, very small amounts. We frequently, when collaborators contact us and they send us samples, we frequently get lifetime supplies. We could use, if they were stable, we could use them forever.
Okay. How long does it take to acquire a movie for mass measurement? I can't remember if you said that already.
No, I haven't. So we usually acquire for about 1 to 2 minutes, but in most cases, 30 seconds of data is plenty to get a nice histogram, get a nice mass distribution.
All right. And how do you know that your sample will attach to the glass?
Well, proteins are sticky.
Sure.
Non-specific interactions, it's quite tough to make a surface unless you charge it significantly, where you're in a situation where pretty much anything will bind. Okay. So we use just standard cover glass, wash it a little bit, clean it a little bit, and so far we've struggled to find something that just absolutely refuses to bind. You might see some variation in the binding strength, but most stuff sticks.
Is there ever a circumstance where, for experimental purposes, you need to change the surface for the measurement?
Yeah, and we do that frequently. So sometimes you want to charge the surface to increase the interaction. Sometimes you want to passivate the surface and functionalize it with something specific, and you can do that. So a lot of the standard protocols that people have used for a long time for surface functionalization, like pegylation, work perfectly well with this approach as well.
Oh, all right, that's great. Well, that is the end of all of our questions. So thanks again, Philip, for that really illuminating presentation. That's some really amazing technology that you have developed. It's pretty cool, and also a great discussion. So, and then obviously thanks to Refine. Um, and thanks to the audience for taking the time to attend and listen. And if you've enjoyed this seminar, and you'd like to view the video recording of the session, you can visit our webinars page at bitesizebio.com, and it'll be available there in about the next 24 hours. And you can also check out some other webinars that we have lined up for you and those that we have available for on-demand viewing. So until next time, good luck to everyone in your research, and goodbye to all of us at Refine and Bite Size Bio.